Following video will cover information about the eukaryotes in subtopic 1.2. The following is a list of understandings, application, and skills that will be covered in this video. In particular, we'll go through drawing the ultrastructure of eukaryotic cells based on electron micrographs, and how to identify organelles based on interpretations of electron micrographs. So there are two main types of organisms, and they are grouped according to their cell type. There are eukaryotic cells and prokaryotic cells. Eukaryotic cells contain a nucleus that holds the DNA, and prokaryotic cells do not contain a nucleus. There are many other features that distinguish the two cell types, but this is the main feature. So in eukaryotic cells, they do have more complicated internal structures than prokaryotic cells, and they contain membrane-bound organelles, meaning that there is a membrane surrounding the particular organelle. And each organelle will have a distinct structure and function associated with it. So there are advantages of having membrane-bound organelles. The enzymes, which are proteins that will perform a particular reaction, and substrates, which are molecules that enzymes will act on. Both enzymes and substrates for that particular process can be more concentrated than if they were spread throughout the cytoplasm, meaning you can have these enzymes and their molecules in a closer area within that membrane-bound organelle than if they were just found floating throughout the cytoplasm. Substances that could cause damage to the cell can be kept inside the membrane of an organelle, and conditions such as pH can be maintained at an ideal level for a particular process. So within some membrane-bound organelles, the pH may be a little bit more acidic, which if if that acidic fluid is released into the cytoplasm, that could cause some damage to the cell. Organelles with their contents can also be moved around the cell as needed. So when drawing the ultrastructure of eukaryotic cells based on electron micrographs, your drawing should include the following if observed in that micrograph. But if you're drawing just based on the structures that a cell would have, then it should include the following list of organelles. So there should be a plasma membrane, a nucleus, a rough endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria with cristae, free ribosomes, a chloroplast, vacuoles and vesicles. If it's a specialized cell that you're being asked to draw, then it should contain cilia or flagella, and if it's a plant cell, it should contain a cell wall. So we'll go through a list of organelles, describe their function and structure. We'll start with the plasma membrane. So the plasma membrane controls what enters and leaves the cell. The membrane is composed of a double layer of water-repellent molecules. So in the following diagram shows that cell membrane. So the two layers of water-repellent molecules, we found that if we draw a line right down the red part of our diagram of the cell membrane, the red lines with the black spheres, all of those would be considered one layer. And then just below that, you have another layer of black spheres with red squiggly lines. And this would be the other layer of the membrane. So taken together, this creates one membrane. And then embedded within the membrane are these large globular type molecules, which are proteins, which can act as receptors to the outer surface, which will detect signals to the cell and then relay these to the interior. So going back to our cell diagram, if we consider this area here, the outside of the cell, and then this bottom layer here, connected to the cytoplasm of the cell. So this would be the inside of the cell. Then if there are molecules on the outside which can interact with that receptor protein, then this could relay a signal to the inside of the cell, providing it with information or instructions. The membrane will also have pores that run from the cytoplasm to the surrounding fluid, so from the inside to the outside. With the nucleus is a double membrane with pores, meaning that it has two membranes surrounding it. And inside the nucleus, this will contain chromosomes. So the chromosomes are the DNA of the cell that are associated with a particular type of protein referred to as a histone protein. In the diagram, the DNA is represented by these blue wavy lines, and then the histone proteins are these yellow cylinders. And so the DNA will wrap itself around the histone protein to form these chromosomes. So you'll have a whole bunch of histone proteins with the DNA wound around them to form those chromosomes. Now, sometimes the DNA will have to uncoil, and uncoiled chromosomes in the nucleus have a special name or a different name referred to as chromatin. So the function of the nucleus then is to act as a site of DNA replication. So this is where DNA will get copied, and in a separate process, DNA is going to be used to create a molecule referred to as mRNA. So the DNA will hold the information for what protein needs to be made. A molecule is made based on the DNA called RNA that holds a copy of the information to build that protein. The mRNA is then used to make the protein with ribosomes. That mRNA that is created within the nucleus is exported via the nuclear pores to the cytoplasm. And don't forget that some cells are multinucleated, which includes striated muscle cells and hyphae. Looking at the structure of the nucleus, so there is the nuclear envelope, which can be considered the purple structure around the diagram. Remember, that is a double membrane, so it would literally consist of one membrane, and then surrounding it would be a second membrane. And then there are gaps in the membrane, which are the nuclear pores. This will allow material to exit the nucleus or potentially even enter into the nucleus. And then there is the DNA that is stored within the nucleus. So within this diagram, it's represented by the orange dome-like structures and the red stringy lines coming off of those orange domes. So the orange portion of the chromosome, or what is represented as DNA, is DNA that is not actively being used. So this is DNA that is wound up. And then the red wavy lines would represent DNA that is actively being used by the cell to create proteins. In electron micrographs, the nucleus would appear generally as a circular structure, and there will be areas where there is a darker portion to the picture, or a darker stain, and areas with a lighter stain to them. So the areas with the lighter stain represent the DNA that is actively being used, while the darker regions represent DNA that is not actively being used. The next organelle is the rough endoplasmic reticulum. And the rough endoplasmic reticulum consists of flattened membrane sacs referred to as cisternae. Attached to those cisternae are 80S ribosomes. And in eukaryotic cells, we denote those ribosomes as being 80S. It just has to do with the size of the ribosome. And this distinguishes these ribosomes from ribosomes of prokaryotic cells, which are 70S. The function of the rough endoplasmic reticulum is that it'll be involved in protein synthesis. And so those proteins are going to be made on the ribosome of the rough endoplasmic reticulum. Those proteins, as they are created, will be passed into the cisternae and then carried by vesicles, which bud off and are moved to the Golgi apparatus. So where we would find rough endoplasmic reticulum in the cell is generally associated very closely with the nucleus. So if that outline in blue is the nucleus, so this would be inside the nucleus, and then you have this portion here outside the nucleus, which would be the cytoplasm. So just outside of the nucleus is where you would find the rough endoplasmic reticulum. And it'll form these series of channels around the nucleus, attached to the membrane, to that endoplasmic reticulum, are going to be ribosomes. And those ribosomes will be attached on the cytoplasm side of the membrane. As those ribosomes synthesize proteins, those proteins will be passed into the membrane itself. So the protein will go into the channel of the endoplasmic reticulum. And then from there, because this is a membrane, you can get vesicles that form from that rough endoplasmic reticulum, which are then transported to the Golgi apparatus. Looking at electron micrographs of rough endoplasmic reticulum, they would appear as a series of channels that appear empty on the inside, and then on the outside surrounding them you would see all these black dots. Those black dots would be the ribosomes. And usually you would find the rough endoplasmic reticulum very close to the nucleus of the cell. The next organelle we'll consider is the Golgi apparatus. So the Golgi apparatus as well consists of flattened membrane sacs, also referred to as cisternae. So the function of the Golgi apparatus is to process proteins brought in vesicles from the rough endoplasmic reticulum. So once these proteins are brought to the Golgi apparatus, they will get processed by adding on carbohydrates or sugars onto the protein, or lipid components if they are to be lipoproteins. So from the Golgi apparatus, once those proteins have been modified, they are then carried in further vesicles to the plasma membrane for release or secretion. So you can have these vesicles that are coming from the rough endoplasmic reticulum that will then merge with the Golgi apparatus. And the Golgi apparatus, again, is these flattened series of sacs. And there are a bunch of them just kind of stacked on top of each other. There's anywhere between 3 to 6 of these membranes stacked on top of each other. In the Golgi apparatus, this is where the protein processing happens. And then from the Golgi, you get the release of these vesicles, which then go to the plasma membrane. Electron micrographs of the Golgi apparatus will appear just as that, as a series of flattened membrane stacks with vesicles surrounding them. The next organelle we'll consider is the lysosome. So lysosomes can form from the Golgi apparatus along with vesicles. Vesicles generally will get released. Lysosomes will stay within the cell. So a lysosome will contain hydrolytic enzymes, which are enzymes that will just break down big molecules into smaller molecules. So they'll break down DNA into nucleic acids, they'll break down proteins into amino acids, etc. So the functions of lysozymes is that they will digest old organelles, so organelles that need to be replaced, they can break them down. If it's an immune cell that has taken up the bacteria, those lysosomes will digest that bacteria as well as viruses. And then they'll also function in the breakdown of biomolecules, including those proteins, nucleic acids, carbohydrates, lipids, and even cellular debris. When looking at electron micrographs of lysozymes, they will appear circular in nature and will have a dark coloration at the center of them. That dark coloration or dark staining indicates that there's a high concentration of proteins within them. The next organelle is the mitochondria, and the mitochondria is surrounded by a double membrane, meaning that if we look at our diagram of the mitochondria, there is an outer membrane that surrounds the whole organelle, and then within that membrane is another membrane that folds upon itself on the inside. So this is that second membrane. So there's an outer membrane and an inner membrane. And the inner membrane is invaginated or folded to form those cristae. And then within the cristae itself, or just outside of those folds, is a fluid referred to as the matrix of the mitochondria. So the function of the mitochondria then is to produce ATP for the cell by a process referred to as cellular respiration, and in particular aerobic cellular respiration. And within the matrix, so within that fluid on the inside of that inner membrane, is going to be the site of a process referred to as the Krebs cycle, which is part of the process of making ATP for the cell. And then the third process, or the last process of making ATP, called oxidative phosphorylation, which is just a fancy way of saying ATP synthesis, occurs on the cristae of the membrane. So the inner folds, or the folds of that inner membrane, are where ATP synthesis will occur. For electron micrographs of mitochondria, they will appear as having this bean shape to them. And then on the inside of this bean shape will be a series of lines which would represent the inner membrane of the mitochondria. The next organelle are free ribosomes. So free ribosomes are not surrounded by a membrane. They are free-floating within the cytoplasm. So when they synthesize protein, they release them directly into the cytoplasm. This is different from the ribosomes associated with rough endoplasmic reticulum, that when those ribosomes make their protein, they release them into the membrane itself. Free ribosomes produce proteins that go out into the cytoplasm. And all ribosomes are constructed in the area in the nucleus referred to as the nucleolus. Ribosomes are composed of two parts. There is a large subunit and a small subunit. So the large subunit is just going to be a larger molecule. The small subunit is obviously a smaller molecule. Together, the two parts come together to make up a ribosome. So in plants only, we find that organelle referred to as chloroplast, which is going to be the site of photosynthesis in those plants. So in the chloroplast, this is where light will get trapped by chlorophyll pigments, which are found in these disc-like structures called thylakoids. So chloroplasts are also double membrane organelles, so they have an outer membrane and an inner membrane. The inner membrane is not folded like mitochondria, but instead chloroplasts have these disc-like structures, or these stacks of discs, referred to as thylakoids. Carbohydrates are made using trapped energy and carbon dioxide in the stroma. So the stroma is that fluid found on the inner membrane. And as a result of photosynthesis, or that process of photosynthesis, oxygen is produced as a waste gas from this process and gets released. In electron micrographs of chloroplasts, they too appear as a bean-like structure, but within the chloroplast you'll have these areas that are very darkly stained that have this disc-like structure to them, or this kind of square or rectangular shape to them. These dark areas represent starch that is being made, or sugars that are being made by those thylakoids, and so appear as a darker stain or a darker color. Next organelle we'll deal with are vacuoles and vesicles. These are single membrane organelles with fluid inside. So the way to kind of think about it is, if we have our cell membrane, we can have a vesicle which is separated from the cytoplasm of the cell, and inside that vesicle will be a fluid that is distinct or separate from that cytoplasm. And then the same thing with vacuoles as well. So plants will typically have a large vacuole. In animal cells, those vacuoles will be relatively small. So the function of the vacuole is that some animals, in some animal cells, it will allow some nutrients to be digested, but it's mostly used for storage of amino acids and sugars. With the function of vesicles is that they will transport materials from one area of the cell to another. We're looking at electron micrographs. Vacuoles in plant cells will appear rather large and will occupy most of the space within a cell. The vesicles will be a lot smaller and will have a dark stain to them. Other structures that are good to be aware of include microtubules and centrioles, which are involved in the process of mitosis. So microtubules are small cylindrical fibers. The function of the microtubule is to move chromosomes during cell division. So after DNA division, those chromosomes will line up in the middle of the cell, and then they will get pulled to the poles of the cell by those microtubules. Those microtubules are attached to centrioles, which form an anchor point for those microtubules during cell division. So those microtubules will then attract, pulling those chromosomes to opposite poles of the cell, or opposite sides of the cell. In specialized cells, there can be certain additional structures, such as cilia and flagella. So flagella are those whip-like structures projecting from the cell surface. The flagella are generally larger and will only usually be present in very specialized cells, such as sperm. Cilia are a lot smaller and may be present, and many are present on stomach cells, for example, and respiratory cells. So they can function, so cilia will help function in locomotion for some cells, depending on the cell type, or can be used to create a current, or wave-like motion that they can form can help move things along. In plant cells, surrounding that cell membrane will be the cell wall, and the cell wall will function to support plant tissue. And the cell wall is composed of a material or a molecule referred to as cellulose, which is a permeable molecule. Cellulose is also known as fiber, so when you're told to eat more fiber, you're basically eating more cellulose. And so that cell wall is also important in helping establish the turgidity of the plant. So it's what makes it, it's what makes celery crunchy or a carrot crunchy, for example. So, in electron micrographs, you can't really distinguish the cell wall from the cell membrane, but the cell wall, will usually be found on the outside of that cell, should you have that in the picture itself. You will also be required to label animal cells and the various organelles within them. That will be covered in class. And you should be able to label plant cells as well, also covered in class. So the following chart goes through some of the big differences between animal cells and plant cells. So plant cells will contain a cell wall, where animal cells do not. Both animal cells and plant cells do have a cell membrane. Plant cells will only have a chloroplast. They are not present in animal cells. Vacuoles will be large within plant cells, and in animal cells, they're not usually present, but sometimes you can find small temporary vacuoles. And plant cells will generally have a fixed shape, whereas animal cells are able to change shape and are usually round in nature. Between eukaryotic cells and prokaryotic cells, there are some main differences. Prokaryotic cells tend to be small, whereas eukaryotic cells will be a lot larger. So prokaryotic cells tend to be around 1 micrometer in size. Eukaryotic cells, can be as small as 10 micrometers, but can be as large as like 30 micrometers. Prokaryotic cells are typically going to be unicellular. Eukaryotic cells can be unicellular, but many eukaryotic cells are found in multicellular organisms. Well, all multicellular organisms will be eukaryotic. In prokaryotic cells, there is no nucleus. The DNA is going to be looped and naked, meaning there are no proteins associated with it, with that DNA, and it's found in a region referred to as the nucleoid. Whereas in eukaryotic cells, the nucleus, the eukaryotic cell will contain a nucleus that contains linear DNA that is associated with histone proteins. Prokaryotic cells will not contain membrane-bound organelles, whereas eukaryotic cells do contain membrane-bound organelles. Prokaryotic cells will have a smaller 70S ribosome as compared to the larger 80S ribosome of eukaryotic cells. Eukaryotic cells will contain mitochondria, prokaryotic cells do not. And the eukaryotic cells will divide by mitosis, or a process referred to as meiosis, whereas prokaryotic cells will divide by that process of binary fission. So that does it for this video about eukaryotic cells.